Hello folks, today we're going to talk about this one of the largest molecules or biomolecules that we have in every cell, how it was being discovered, the structure and its role. This is the three-dimensional structure of deoxyribonucleic acid or the DNA. This is another nucleic acid called ribonucleic acid or RNA, which is usually single-stranded, and we all know DNA happens to be the carrier of genetic material. With a few exceptions of viruses, they may or may not have DNA, they could have RNA. Now, the question was, long ago, is DNA the genetic material? And if not, why not? So, there were a large number of experiments being performed to prove the same. The search went on, and it was found from the studies of viruses and bacteria. Yes, the DNA does play a role in affecting them, and the experiment of Frederick Griffith on bacteria Streptococcus paved the way to prove that yes, DNA do have genetic material in it, or rather the genetic material is composed of the DNA. Let's begin. What's Griffith's experiment? We learned already that yes, there are S forms, they are virulent forms, they have the capsules, and hence this body cells cannot fight it out or have their defense mechanism against it. And once injected into a rat or a mice, it dies because it is virulent, capsulated. Let's take another strain of bacteria, R forms. On injecting, we found the animals to sustain because they were non-virulent. They did not have a capsule. Now, capsule happens to be the outer covering, as we all know. Now, if the S forms were killed and destroyed, obviously we are trying to destroy the outer layer, that is the capsule, and then inject it to an animal. We expect the animal to live because the capsules have been removed, which were preventing them from being destroyed by the body cells. Now, in another experiment, the same killed S forms and the R forms were taken and injected to animals. Obviously, we expect nothing to happen because the S forms are non-virulent and we are taking the killed S forms. R forms are non-virulent and we are taking the killed S forms together. Now, when these were injected together, we expect nothing to happen, but here animals died. Now the question was, why did they die? What happened? And then they also found the body of the dead animal contained S forms. Now, how did this one come? We never injected these kinds of S forms, capsulated forms. In fact, we had killed them. Now, what might have happened was the R forms got transformed into S forms, and that killed them. Now, that means the S forms, when heated, retain the DNA content, nothing happened to them, but their capsule was being destroyed, and therefore the DNA of S forms transformed the R forms and thereby produced the capsules, and they became virulent and hence killed the animal. Wow, that was a very brilliant experiment to prove that yes, it is the DNA which passes on from one species or one bacteria to another, and they are the genetic carriers or inherit carriers, or they carry DNA. And that was it, transformation. Further evidences for this being the genetic material came forward, and lots of studies on viruses that infect bacteria, that is referred to as bacteriophage, were used and came into light. Aman, would you like to throw some light on Alfred Hershey and Martha Chay's experiment?
Well, they selected the T4 bacteriophages, which generally attack Escherichia coli, and there are actually T1, T2, T3, T4. All have two kind of a structure, that is, there is a nucleocapsid and a tail. Now this nucleocapsid region has DNA inside, and the outer coat, that is capsid, is rich in protein. We know protein have always sulfur in them, so they use that particular feature, and they use radioactive material for proving that viruses also have DNA, which is the genetic carrier.
This is another experiment of Avery, McCarty and MacLeod.
So these people in fact wanted to prove Griffith's experiment. So they picked up the same strains of Streptococcus pneumoniae, the smooth forms and the R form. Here the smooth forms were the ones which were heat killed. So the smooth heat kill forms were treated with various enzymes and then mixed with the living R form and then cultured on the petri dish. So in the first case, the enzyme used was proteinase,
which could dissolve proteins,
and the heat killed S forms when treated with proteinase and then mixed with the R living form and then put on the petri dish. It was amazing that the daughter colonies which were formed, they were having the smooth living forms in them.
the virulent forms,
Yeah,
which we call
virulent forms. So in the second case, they mixed the heat killed S form with nuclease enzyme,
which would have dissolved
which could have destroyed their DNA.
Okay.
And then they mixed it with the R form, that is a rough non-virulent form. And
then they
allowed it to culture on the petri dish, and they observed that the whole colony was only of the rough form. That means it's the DNA which is the genetic material, and once we destroy that DNA, it cannot result in transformation.
Wow. Well, this is the Hershey Chase experiment. In fact, they took viruses and they tagged with some radioactive elements. Now, and they took radioactive phosphorus as well as radioactive sulfur. Now we know that sulfur would be picked up outside, because the outside covering or the capsids are usually made up of proteins, and proteins contain sulfur. Again, the phosphorus labeled radioactive elements would have been within the viruses, that is inside the DNA, because the DNA do contain what we call the phosphorus groups. Now, with these tagged radioactive elements, these viruses were led to attack the bacteria, and subsequently what the researchers found was that these viruses, when infect, get into it, leaving certain entities, that is the coat outside, and inside it they introduce their genetic material, the DNA, and that is used up by the bacteria, as we all know, and thus lot of viruses, daughter viruses, are formed which come out. On analysis, they found that the new viruses had, okay, why don't I ask what was the result to Aman?
Well, as we know, the capsid was left outside, and it was only the DNA which was pushed onto bacteria to form the daughter virion. So all the daughter cells of the viruses, they were having capsule which was without any sulfur in it, whereas their genetic material all was having radioactive phosphorus. Thereby it was a strong proof that proteins have nothing to do in the participation of carrying on the genes. It is only the DNA which is genetic material.
That was a wonderful experiment again.
And because these people, they used a blender so as to shake the bacteriophages.
One second, let's go to the next slide to show them.
Okay.
Here, if you see,
to shake or the phages,
agitate, yes.
They used blenders. Therefore, this Hershey chase experiment is very famous on the name of Blender's experiment.
And in fact, they got this blender from their neighborhood.
Yeah, because they were not in their lab and they had gone for their vacation somewhere, but they wanted to perform this experiment.
What a famous Blender's experiment of Hershey and Chase. Now, how does the genetic material look like? The DNA particularly, discovered long ago, and it was Watson and Crick who deciphered the same, along with X-ray analysis of Rosalind Franklin. In fact, they had gone for an evening dinner with her when they discussed the structure of DNA. The DNA molecule, of course, is usually seen to be in a coiled or supercoiled structure in chromosomes, and the chromosomes stay inside the nucleus of a cell. And we have found the 3 billion DNA subunits in form of their bases, and 30,000 genes, approximately 46 chromosomes of human body. And it is a double helix, 2 strands coiled to each other, containing sugar, phosphoric group, or phosphoric acid, and nitrogenous bases. The bases are in the middle, if you are able to see in green, stuck with sugar molecules in gray, and then attached to phosphorus. Now these 3 components together form the nucleotides. So you have series of nucleotides on one end, on one side, onto the left, and you have series of such nucleotides on the right. So you have 2 chains of polynucleotides. So these 2 chains of polynucleotides are held together between the bonds formed between the bases, that is, and they are hydrogen bonds. The kinds of bases that we observe are 2, purines and pyrimidines. Now, the pyrimidines are single ringed structures, that is on the first row as it is shown here. They are 3 kinds: cytosine, uracil, and thymine. Uracil is exclusively found in RNA and not in DNA, whereas thymine is found in DNA and not in RNA. So, in a way, we have 3 kinds of purines, and pyrimidines, I'm sorry. And you have purines, two kinds, adenine and guanine. They are referred by A and G, and they are double ring structures. Well, this is again the same thing that we discussed a while ago. And we also know nucleosides to contain sugar and a base. Together they form what we call nucleosides, and there are bonds between them called beta N glycosidic linkages. We're going to see that. But this is a nucleotide, the phosphate group, sugar and a base, and that is the detailed structure of it. The sugar could be a ribose sugar, and nitrogenous bases containing primarily nitrogen compounds. And here we see how they are structured and how the phosphate looks like, how the sugar looks like, and how the bases are, and what the bases are. The bondings in the DNA, as it is referred here, between phosphorus and sugar and the bases, and you have several kind of linkages, 3' to 5' phosphodiester bonds by which the DNA is linked, and these bonds are referred as 3', 5' because every sugar molecule has carbon which are designated as 1, 2, 3, 4 and 5, and if the third carbon attaches at one end with another molecule, that end is referred to as 3', and the carbon molecule at fifth position, if they are bonded to another molecule, the at other end is referred to as 5', and hence every time you have a 3' end and a 5' end where the bonds are being formed. Let's have a look at those phosphodiester linkages. Here, with an arrow, it has been designated to happen between the sugar and a phosphate bonds, and you have another kind of bond between sugar and the bases that is called N-glycosidic linkages bonds. So this polynucleotide polymer is one of them, and such two of these, they join together to form what we call the DNA. Let's have a little practice. You have to name the structures labeled as 1, 2, 3, 4, etc., etc. By now, you might have got familiarized with some of the components of DNA. These are some interesting aspects to the same molecule, how enormous they are. Let's talk about the hydrogen bonds. Aman, could you throw some light on the hydrogen bonds formed between the bases?
Well, when the two strands of the DNA, they try to fit with each other to form helix, the bases in between them, pyrimidines always get attached to purine and vice versa. That is, pyrimidine of one DNA strand will get attached to purine of another DNA strand.
So,
through the bondages, which is called hydrogen bonding.
A would always go with
Thymidine.
And that is adenine with thymidine, and G that would always go with C. That is
guanine will always get attached to cytosine, but there is a difference here. Whenever A gets attached to T, there is double bond. Whenever C gets attached to G, there are triple bonding.
we know that the DNA is a double helix structure. The two strands, they're coiled and they're bounded to each other in form of a spiral staircase. That is the one that you are able to see. On the right is a 3D marker. The purine bases are always attached with pyrimidines by hydrogen bonds. Yes, we discussed all these issues a while ago. The same here. The hydrogen bonds hold the bases. The details yes, if one DNA strand on the left happens to run from 5' to 3', then obviously to fit in the other strand on the right with hydrogen bonds, it has to be from 5' to 3'. So one, if it happens to be 3' to 5, the other would be 5' to 3, or the vice versa. And such type of two strands in two different directions held together are referred to as anti-parallel band strands. Let's have a look at it. Here we see the two strands are anti-parallels. Usually, a DNA would have, for every turn or every helix, a set of nucleotides or bases between them. Usually we find 10 to 11 and sometimes 12 bases. So the distance that one helix completes is about 3.4 nanometers between the bases. It would be therefore reduced to 0.34 nanometers, considering that there are 10 base pairs between them. The RNA molecules contain uracil instead of thymine. They have ribose sugar and they are usually single-stranded, and these three are important and unique characteristics of RNA molecules. There are three kinds.
Well, the three kinds of RNA, we have transfer RNA, tRNA, we have rRNA, that is ribosomal RNA, and the third one, messenger RNA. tRNA, it is used to carry amino acids to the site of protein synthesis. rRNA helps in packaging of ribosome, and the third one, messenger RNA, is only formed on DNA at the time of protein synthesis through process transcription.
The details we will be going through later. Here we see a RNA molecule, single-stranded structure, and also we have what you call U instead of T in this.
It's 8 o'clock.
In this particular molecule. These are the components again of an RNA molecule: the phosphorus, the sugar, and the base. Same way as we have it in DNA, except that it is a single strand. the uracil replaces the thymine in case of RNA. The base pairing is the similar way, adenine with uracil and guanine with cytosine. Since they have only one base pair and not bonded with any hydrogen bonds, so they sometimes form a coiled structure and reattach their bases amongst each other and giving a 3D structure, which we are going to have a look at it. Three kinds of RNA molecules: ribosomal, messenger, and transfer. We see a transfer molecule, a transfer RNA, the mRNA, and the white colored RNA molecule within the ribosomal units, that is given in gray, called ribosomal RNA. Could you say something here about messenger RNA?
So as I was talking about before too, that messenger RNA will be synthesized only at the time of protein synthesis on one strand of DNA, and the process is referred to as transcription, and then moved on to cytoplasm. Ribosomal RNA, it helps in packaging the ribosome, and this packaging is done inside the nucleolus in the nucleus, and then they are passed on to the cytoplasm, and it is about 75% of RNA found in the cell, most abundant kind of RNA. The transfer RNA is called transfer RNA because it is a carrier molecule to pick up amino acid to the site of protein synthesis. That is, it brings them to messenger RNA, and it gives a shape due to attachment of its own bases, which is called clover leaf, having two specific ends, 3' end. We will just see in an image ahead. So the 3' end is the one where amino acids are attached. It is called amino acid attachment site. And another site important is at the base we can see the three bases. This is called as anti-codon site, which will be forming a specific hydrogen temporary bonding with messenger RNA molecule with their codons. Codons are always, one codon is made up of 3 nucleotides, therefore anti-codon is also always made up of 3 nucleotides.
Now the big question is, how does the DNA replicate? Replication is different from duplication. In a process duplication, you have 2 units. One could be totally, absolutely new, not having any of the entities of the parent one. But we call this process as a replication because in the next stage of division, you always find some portion of the parent, and hence replication. See, what's the basic principle? Now, the DNA strands, they split apart by uncoiling themselves, and the 2 polynucleotide strands are now referred to as parent one. They give place so as to form the new ones, and the new strands that are being formed on the 2 strands of the parent DNA are called complementary strands. Here you see the same. The parent strand splits, the complementary strands are formed over it. So obviously you have 2 complementary strands on the 2 parental strands which have split apart. Now, how the DNA replicates with many of the enzymes, that is the proteins. Let's have a look at it. The first thing, the DNA unwinds, and the base pairs look something what we are seeing here on the left. the base pair splits apart, the hydrogen bonds are gone, new nucleotides add up, and thus two complementary strands are formed. Now, this looks simple. But before we go, let's learn what is the theory about semi-conservative replication, or what does it mean. There are three kinds: dispersive, semi-conservative, and conservative phenomena.
These three kinds were actually hypothesized by famous scientists Meselson and Stahl, who proved ultimately that DNA replication is semi-conservative type. So the two hypotheses of conservative and dispersive, they were proved wrong, because if it was conservative, then the dotted strands formed, both strands should have the parental strand as such, but it was not like that. And if it was dispersive, then part of the nucleotides should be the mixture of the new and the old, even that was not like that. Every time in the daughter strands, one strand belonged to the parent and one strand was completely synthesized new.
And thus semi-conservative.
That means parental characters were semi-conserved.
This was this experiment.
So here they used some radioisotopes of nitrogen. One is called heavy radioisotope N15, which was used to stain the parental strands, and then parental strands were shifted in a media which was rich in the light nitrogen isotopes, that is N14. Then they were allowed to replicate, and after they were centrifuged in cesium chloride, the results were found, and the results proved that it is a semi-conservative type of replication,
which occurs in DNA. This is not the way it happened.
Yeah, because the strands which were visible of DNA after centrifugation, it proved that it is the one strand totally contributed by N14, synthesized on N15, that is a parental strand. So the band was visible in the center, the upper one, the light strand, and the lower one, the heavy strand. But in this conservative, the result was not such.
Let's have a look at it, what happened in dispersive phenomena.
Again, dispersive, the way it should have been, that it should have shown the mixture of light and a heavy nitrogen radioisotope. Again, this was not the result, what they found.
And ultimately they found.
So what they traced was this. That is the upper, when they observed the first generation, the one strand was parental, one was daughter. When they observed the second generation of the same, they observed that
new strands
altogether new daughter strand, the lighter one, but the other one parent strand was always there in the generation of the DNA.
It's 9 o'clock.
So DNA replication is very unique, and it has a remarkable speed and accuracy. Let's go through it. The first, there is an enzyme called helicase. Obviously, name suggests everything. It unwinds the two DNA strands. So the unzipping breaks the hydrogen bonds, and the two template strands are exposed, that is the parental ones. Once this happens, there is a fork formation, what we call the replication fork. This is how it looks like. The enzyme helicase attached at the base of the fork, trying to break the bonds between the bases. The helicase acts at different sites of a DNA strands, and therefore there are bubbles which is exhibited here. In fact, the bubbles come out because of the reason that the two strands swell up and break up the hydrogen bonds at different places.
This helps, in fact, to increase the rate of replication.
Of course, ATP is useful in this process.
Because replication is an energy-driven process.
There is another enzyme.
Now, once helicase has opened the two strands, but each strand is further supercoiled too. So we have to remove those supercoils also to stretch it. And that stretching,
or rather uncoiling
it, that process is done by this another enzyme, gyrase, which belong to a very big class of enzymes called as topoisomerases.
What are SSB proteins?
Once the bases they are exposed, there is a danger that some other mutant can get attached to them, thereby causing mutation in DNA. To prevent that, the open bases are always capped by certain proteins which are called single strand binding protein.
Are they
SSB?
Are they temporary sealants?
Temporary sealant we can call them.
Now, what are primers?
Well, DNA polymerase is the enzyme which has to carry on the process of replication, that is bringing a respective nucleotide to the parental strand. Suppose there is E, it is going to pick up the T nucleotide. But DNA polymerase cannot recognize the replication fork. This replication fork is recognized by another enzyme, which is a class of RNA polymerase, referred to as primase. So first, primase brings its bases, but as we know, RNA polymerase cannot bring pyrimidine. Instead, it brings uracil. Therefore, 5 to 6 bases, which are attached in the beginning on a DNA strand, they are the bases of RNA, and that structure attached to DNA is referred to as primer. Primer is recognized by DNA polymerase, and rest of the process is then carried on by DNA polymerase to complete the DNA replication.
Let's have a look at it.
Now there is another feature to it, that the two strands of DNA, one is called as a leading strand, which will be requiring only the one which is going to move from 5' to 3'N. It will require only one primer, and the other one is a lagging strand, that is from 3' to 5'. It will be having multiple primers.
The leading strand formation, is it a continuous one?
It's a continuous process. But
Whereas
the lagging strand is a discontinuous. It synthesizes 1000 to 2000 nucleotide, referred to as Okazaki segment. Again replication stops, a new primer is added, and again another Okazaki is formed, and maybe another and another. That is, multiple primers will be there on the lagging strand.
Let's have a look at the structure, how the leading and the discontinuous strands are being formed. Here it is.
So in this case,
The upper one.
Yeah, it is towards the fork which is a continuous replication, and the one which is going away from the fork, it is a discontinuous replication. So the one which is discontinuous, it will be having multiple primers, and the one continuous, we will attach primer in the beginning, only one, and then rest it will be.
And these small bits of segments or complementary strands that have been formed are referred to as Okazaki fragments, which will be joined
by the enzyme ligase later on.
Okay, here it is.
So you see in the leading strand we have only this purple color structure, one primer, whereas in the lagging strand you
on
can
the left
trace here on the left side there are two primers attached.
and these small strands will be joined now.
So it's continuous on the 5' to 3', which is leading strand, but 3' to 5' will be the lagging strand.
This aspect is very important, that the leading strand happens to be from 5' to 3' end. Let's have a look at this animation. The helicase comes in, unwinds, the DNA polymerase attachments.
So here the red colored structure, see the primer is attached, but because it is a lagging strand, we will require another primer here.
Whereas on the top you have the leading
Only
strands.
one primer in the beginning, and then it is a continuous process.
So the DNA polymerase helps in formation of the strands.
Yeah, see in the lagging strand, these red colored, see here 2 primers have been added.
And the DNA helicase continues to uncoil.
So another primer comes, that means in between there are Okazaki segments.
So here another one has been added.
So here, another thing is very clear from this particular animation, that every time any DNA replication, in the beginning it will be having certain bases belonging to RNA which have to be removed. There is a proofreading enzyme which will remove these primers, that is DNA polymerase 1. It will remove the primer, and then ligase will attach them so as to complete the DNA.
Let's have a look at it, and you point that tissue at that point. Leading strand on the top, lagging strands being formed at the bottom.
So it's visible there on the top in leading strand if there is a 3' end where the DNA replication starts. Actually, there is a reason to it, because DNA polymerase in any case can only get attached to the 3' end. It cannot start replication from 5' end. So once it is attached to the 3' end, naturally the daughter strand form will be the 5' to 3'. That's why we say continuous DNA strand is 5' to 3', and discontinuous will be from the other side.
True, this was the trick you explained.
So we can go further.
The short fragments called Okazaki now will be attached together with the enzyme ligase, or DNA ligase, one might call it. And that completes the formation of the strands.
See here again, this figure is very clearly showing that 5' to 3' daughter DNA strand is a leading strand and it's a complete one, but the other one is this red color structure has again 2 primers shown here. It is a fragmented structure. Later on it will be joined by ligase.
That was all about DNA replication and the structure.